Hello everyone, my name is Sarrazin-Gendron and I'm presenting assembling a database of heterogeneous RNA 3D loop modules from conserved substructures. The software name is Kentaloops, and the main topic here is the classification of RNA 3D modules. They're also known as RNA structural modules, RNA 3D motifs, but really you can name them what you want. I'm interested in classifying recurrent substructures of RNA that we can represent as graphs. So RNA folds hierarchically, right? You have the sequence and the secondary structure and the 3D structure. And these RNA 3D modules insert themselves within and between loops in the secondary structure. And the problem with them is that because they are non-canonical base pairs, we cannot use our energy models to predict them the same way we do for the secondary structure. So we have to use a different method, such as, say, a statistical model, such as the Bayesian network, to predict them in parallel of the usual pipeline for structural prediction. And this comes with one difficulty, which is that modules are actually flexible. You can have a bulged out nucleotide, and that doesn't really change the local 3D organization. So, what does an RNA 3D module looks like? Well, here's an example. It's called the king turn. So this motif is very common in RNAs. It's in almost every single ribosome, and often in multiple copies. And you see here, I showed you three on your screen. And the particularity here is that all three have the same kink, right? It really introduces a sharp turn in the backbone, but all of these are different loops. Like this is an internal loop and this is a junction, and so they're not the same graph, but they have the same function. So what can we do about this? Because we don't want to train different models to learn how to recognize the same thing. And the solution to this is to group similar graphs into families of similar graphs. So to do this, we use Carnaval, a database that identifies common subgraphs in RNAs. And then we take all the graphs that have the same recurrent interaction network, so a common subgraph. And in all of the occurrences, we compress the graph to make it represent mostly the recurrent graph it has in common with the other ones, and also the closing base pairs, and the rest is compressed to one node. So this allows us to start from an ensemble of a bunch of different graphs that have the same recurrent interaction network in it, and then we classify them by size, then we compress them, and then we do an alignment with dynamic programming such that we have a representative for each category so that each node from the representative aligns to one node in each of the graphs of the category. And what that does is that it allows us to train a Bayesian network on just the representative and then predict at the same time all the different motifs shown in pink at the bottom with all the possible bulbs at the same time. And that's key because it really allows us to not train different models to identify different forms of the same module, the same recurrent structure. And what is this used for when well, first, it helps us make better databases because understanding which graphs are similar can help us group motifs better. And instead of having different entries in the database, we can have the same entry for cousin or sibling motifs, and that can help us better understand the function they have in common. And this applies to better predictions as well, because we do have software tools such as base pairing and base pairing 2 to identify these motifs into RNA sequences. And we have other techniques that are being developed to identify these motifs in sequences and then predict which ligands could interact with these motifs and so have a sequence to ligand prediction pipeline. And the better our classification of functional families of motifs are, the better our overall predictions are going to be. So this is really a overall strategy to have a better, more flexible definition of what the RNA module is and how we can build on this to achieve better results for all methods that use this kind of knowledge. Thank you very much and have a great day.